Here, I'll go into more detail on how to approach validation and provide resources and tools to help you get started conducting a validation study. Validation is the scientific evidence supporting the implemented preventive control that will control the identified hazard. Verification is procedures put in place to ensure that the preventive controls are implemented. Validation, in other words, are we doing the right thing to control the hazard? Verification, in other words, are you doing what you said you would do? Validation establishes the scientific basis for process preventive controls in the food safety plan. You do not have to validate sanitation, allergen, or supply chain preventive controls. The scientific basis for the process preventive control can be scientific principles and data, expert opinions, and in-plant observations or experiments. It is important to note that the validation of process preventive controls must be overseen by the preventive controls qualified individual. For the preventive controls for human food rule, the process preventive control ideally should be validated before production, but must be validated within 90 days of production per the regulatory language. Revalidation is the process of making sure that the process preventive control is still valid and that you are still controlling all relevant hazards for your product. You must do this every 3 years, as written in the regulation. You also need to conduct revalidation when your product or process changes, you have significant changes in raw material, you have continuously recurring deviations, new information or hazards have been identified for your product, or for a number of other issues that point to the fact that your process preventive control is not working as intended. Unlike Hazard Analysis and Critical Control Point Regulations, or HACCP, the Preventive Controls for Human Food Rule requires revalidation every 3 years rather than every year. Like HACCP, revalidation is required anytime there is a significant change in the product or process. Here are the 3 steps in pre-validation: identifying hazards needing control, define the food safety outcome required, and select the process preventive control to be validated. Let's talk about the first step, identifying hazards needing control. I'm going to spend a bit more time here talking about hazard analysis. For each step in the process flow, from receiving ingredients to final product leaving the facility, a determination of potential hazards needs to be made. Once hazards have been identified, an evaluation needs to take place to determine if that hazard requires a control. How severe and likely the hazard is one parameter that is evaluated. If the hazard is severe, it must be determined if prerequisite programs like good manufacturing practices or GMPs adequately control that hazard. If the hazard is not addressed by prerequisite programs, then the hazard may require a preventive control in the process. It can be a challenge to determine what types of hazards are likely or reasonably foreseeable to occur in different products. An important first step is gathering data. This can include information on previous recalls or outbreaks in products like yours, scientific reports, or other guidance documents. It is also important to consider not only products exactly like yours, which data may not exist for, but for other products in related categories or even raw ingredients that you are using. Both FDA and USDA post recall information on their websites. You can search by category there, and it is a good place to start. The FDA also published guidance documents to help you figure out what types of hazards are associated with different ingredients and formulated products. The ones shown here, Hazard Analysis and Risk-Based Preventive Control for Human Food Draft Guidance for Industry, contains information on biological, chemical, physical, and process-related hazards associated with specific ingredients or products. To search for hazards in this guidance document, you'll want to use Appendix 1. There are tables for biological hazards, chemical hazards, and process-related hazards. If you open the PDF and hold down the Control button and press F, a Find box will open. You can type the name of the ingredient or product of interest here and press Enter to find sections in the appendix relevant to your product. Another tool that you can use to determine relevant hazards is the International Commission on the Microbial Specifications for Foods. This document will also list hazards associated with specific foods and also provide information on how you might implement a process control for that particular hazard. This resource is divided into two parts. Part 1 focuses on how to use data to validate a microbial process control. Part 2 focuses on how to apply process controls for microbial hazards to different categories of foods. Let's move on to the next step prior to validation, defining the food safety outcome that is required. The food safety outcome is what level of reduction or control of microbial growth is required to maintain the product's safety. This might be predefined as performance criteria for specific products, or you will have to establish your own food safety outcomes based on existing data. Let's take a moment to define microbial inactivation and inhibition of microbial growth. Inactivation refers to a reduction of the number of microbes in a food product. The process used for inactivation is normally called the microbial kill step. The specific level of inactivation required, or the log reduction, of the microbial hazard may be defined by regulations or may need to be determined internally. Inhibition of microbial growth is another way to minimize food safety risks. In this case, the product's formulation or storage conditions are specifically designed to keep the hazardous microorganisms from growing in the food. Examples include formulating a product to reduce its water activity, adding an acid to reduce the product's pH, adding preservatives, or storing a product frozen or refrigerated. This table shows the relationship between a product's pH and water activity and its impact on the potential for microbial pathogens to grow. On the far left column are water activity values, and the top row is the product pH. You can see within the matrix highlighted in green where there is no anticipated growth of pathogens in a food with a particular combination of a water activity and pH value. In products with a water activity less than 0.88, there is no growth of pathogens even when the pH is greater than 5. However, when the water activity is higher, greater than 0.92, you can have growth of pathogens in foods with a pH greater than 4.2. Those combinations where PHF/TCS is listed are potentially hazardous food or temperature controlled for safety. This means another control needs to be implemented, like cold storage, to minimize food safety risks. Another common challenge is determining what level of microbial foodborne pathogen inactivation is required for different products. This slide outlines a few existing regulatory performance standards or levels of bacterial log reductions for different foods. As you can see, there are not many on this list. The log reductions listed here are required by law for these types of foods. For others, you may rely on some of the resources shared earlier in this presentation, other published research, or your own internal data to determine the appropriate log reduction required for your product. When trying to determine your own internal target level of reduction, the International Commission on the Microbial Specifications for Food uses an approach which focuses on what the level of pathogen would be at the consumer stage. It's based on what the anticipated level of pathogen would be in the raw ingredients, what level of reduction would be anticipated during the kill step, but then how the pathogen level may increase during distribution, storage, and final preparation. The level of reduction required at the kill step would then be reassessed, taking into account any anticipated increases at the consumer level. The final pre-validation step is selecting the process preventive control that needs to be validated. There may be more than one process preventive control that needs to be validated, depending on your product and the process you're using to inactivate or inhibit the microbial hazard of concern. To select the process preventive control, it's good to go back to the flow diagram. Think about the mode of action at each step, i.e., does this step inactivate pathogens, or is it intended to inhibit growth of pathogens? You should also assess if there is potential for recontamination at any step, or if there is potential for growth of pathogens in your product after an inactivation step. When selecting a kill step or inhibition process, it is important to know your product's characteristics, such as water activity, pH, fat, and protein content, as well as its physical properties. All of these can impact the method of inactivation or inhibition selected, as well as the parameters needed for proper inactivation or inhibition. This table shows common process preventive controls used for biological hazards in food. Lethal treatments, or kill steps, commonly used include heat treatments, antimicrobial fumigation, high-pressure processing, irradiation, and some others. Inhibition processes include reducing storage temperatures, manipulating formulation to lower water activity, pH, or adding preservatives, and also dehydrating or drying. For lethal heat treatments, here are key pieces of information you'll need for validation. First, what is the heat-induced inactivation profile of your target pathogen? This is referred to as thermal death time data. Next, you will need to know heat transfer or heat penetration data for your product. Knowing these will allow you to ensure that you will reach the appropriate temperature to destroy your target pathogen in your food product. Here, we will describe some common terms that are used to define microbial inactivation in the context of thermal processing. These values are determined by doing a series of experiments to determine the level of inactivation of a particular bacteria in specific media using temperature and time as variables. First, let's introduce decimal reduction value, or D-value. D-value is the time required at a specific temperature in a specific medium needed to reduce the number of a specific microorganism by 90%. Z-value is calculated using a series of D-values calculated at different temperatures. The Z-value is the temperature increase that is required to cause a 90% reduction in a D-value. The thermal death time, or F, is the amount of time required to kill a specific number of target microorganisms at a specific temperature. Determining these values and conducting the calculations to determine them can seem challenging. We'll just give a bit of basic overview of the way we think about bacterial numbers in food. This table shows an initial load of microorganisms in our food of 10,000 or 4 logs. Next, the D reflects how many reductions of base 10 or logs our process is targeting. A D1 value means we get a 10-fold or 90% reduction in the initial population of the microorganism, leaving us with 1,000 or 3 logs of the microorganism remaining in the food. Theoretically, if we apply a 6D process, a process targeted to reduce our target microbe by 6 logs, the resulting product would have 0.01 or log minus 2 bacteria left. Depending on the process being utilized, there are a number of factors that influence the microbial thermal death time. For any thermal process, the heat transfer within the product is a critical factor to evaluate. For traditional thermal processes, whether a product is heated by conduction or convection will influence how quickly and evenly the product reaches the uniform temperature. For other thermal processes, such as microwave, radio frequency, or ohmic heating, there are additional factors to consider. This is why it is important to do an in-plant validation to ensure that you can achieve the desired temperature for the target time in your product. For non-thermal processes, process factors also need to be considered. For high-pressure processing, the time, pressure, and temperature all need to be evaluated for impact on microbial death time. For UV light, the wavelength, time of exposure, and penetration depth are critical factors to evaluate. Whichever process is employed, it's up to you to understand the process factors that need evaluated and establish the critical control point. Again, a brief review of critical limits and operating limits. The critical limit is the value or parameters necessary to control the hazard that was identified. Therefore, if that critical limit is not met for any reason, there is a potential risk to human health. Failing to meet the critical limit results in a deviation from the scheduled process. An operational limit is a value or parameter that is set by a processor that is typically more stringent than the critical limit. It allows for a buffer zone in which failure to meet an operational limit allows for a process adjustment before the process deviates from the critical limit. These process adjustments can be made with less or no documentation, which saves time. Here is a chart illustrating critical and operational limits for a thermal process. The x-axis represents time, and the y-axis represents temperature measured in degrees Fahrenheit. In this example, the critical limit for the process is 160 degrees Fahrenheit, and the operational limit is 180 degrees Fahrenheit. The dark red line represents the measured temperature of the process over time. You can see that there are 2 points where the process falls below the operational limit. In both cases, a process adjustment was made to bring the temperature back above the operational limit. However, there was a third dip in the temperature that fell below the critical limit. In this case, documentation of the deviation and a corrective action were required. For each process preventive control established, appropriate corrective actions are outlined in case a deviation from the set critical limit occurs. For your validation studies, you can rely on existing data and guidance documents. The FDA Seafood and Juice Hazard Guides are useful, as well as the FDA Food Code and its annexes. There may also be useful data from trade organizations. There are also validated microbial modeling programs that can be utilized to determine how a specific microorganism will survive in a specific food under different processing conditions. The USDA's pathogen modeling program is the most commonly utilized. You can also refer to peer-reviewed scientific literature, as many faculty and food science departments work on validation studies for different foods. You may also reach out to University Extension. There are many universities that function as a process authority and can give you specific guidance on your validation study. A consultant may also be a useful tool for developing a validation study if you do not have experience. To validate a process preventive control, you must first decide on the approach you will use. Then you gather validation data and analyze the results. Then finally, you document and review the validation. Let's first focus on how you might decide on your approach for validation. This slide highlights different approaches to validation. You will likely rely on multiple approaches to validate your process. If you are working with a product that falls under safe harbors, or if there are previous validation studies on the same product and process to control for your target pathogen, you would document this information. Then, you would proceed to a plant study, where you verify that you can replicate the process established in your facility. If you have a unique product, you will need to conduct microbial challenge studies. This is where you inoculate your food with the pathogen of concern in laboratory and pilot plant studies to ensure the kill step does in fact inactivate the pathogen. Once you've established the kill step works in these studies, You'd then move to conducting an implant study using a non-pathogen microbe, for example, a surrogate, to show this process does inactivate as anticipated. There are some food products and processes that have specific regulations or guidelines that pertain to their production. These safe harbors are specific times and temperatures for foods that do not require additional microbial inactivation studies. You will still need to do implant operational validation to ensure that your equipment can achieve those specific times and temperatures. There are very few products that have safe harbors. Low acid, shelf stable foods and milk are two examples that have safe harbors. You might also be able to rely on existing scientific studies or technical evidence if the study design mimics your product and process. You'll want to pay close attention to the microorganisms used and how they are inoculated into the matrix. You'll also need to ensure that the matrix used to do the test mimics your food product. If there aren't existing studies for your target pathogen in your food matrix using your process, you'll need to conduct your own challenge study. Here are some important points when conducting lab or pilot plant microbial challenge studies. The study design must mimic your product and process. Make sure you're using the appropriate food matrix and processing treatment. Select the pathogens most likely to occur in your product. Ideally, you'll select strains of that pathogen from outbreaks or that have been isolated from food. You can often buy bacterial strains from the American Tissue Culture Collection, or ATCC. Sometimes universities also have a collection of strains they isolated. Based on the treatment you're using, you'll also want to select a strain of the pathogen that has been shown to be most resistant to that particular treatment. This would make sure your process is effective against the most resistant organisms that might be present. Large food companies have the capacity to do this work in their own laboratories. For a small company, you may work with a third-party lab to conduct these pathogen challenge studies. For some unique or complicated processes, it can be necessary to do implant challenge studies. When conducting an implant study, you never introduce pathogens directly to the processing facility. Surrogate organisms are non-pathogenic microorganisms that have similar growth and inactivation kinetics to the target pathogens. This means they tend to respond to control the same way as the pathogen. Finally, once you've established what the critical limit should be to control your target pathogen, you'll want to do an implant operational validation study. This is to ensure that you can achieve the critical limit in the plant. This implant operational validation could include heat penetration data within your product's matrix, establishing the cold spot during thermal processing, evaluating appropriate belt speed for a continuous process, looking at depth of product on a belt and determining heat penetration, pH measurement, water activity measurements, among many others. Designing an implant study. Every plant is different, so you have to take implant variation into account when designing an implant study. It also has to capture the worst-case scenarios, so when mistakes occur, the validation study is still able to address and cover the issue. There are 3 other bullet points that need to be considered while conducting an implant study: location of sampling site within the process flow, types of samples collected, and method of analysis. Speaking of analysis, all the experiments should have triplicate independent trials representing different lots, shifts, or production days. There are several reasons why 3 independent trials are needed. One is for statistical purposes. Generally, the more variation in a process, the more independent trials and samples that are required. Once you've decided on the approach, the next step is to gather validation data and analyze the results. When you're relying on existing data, your focus will be on the implant operational validation. Again, making sure you can hit those critical limits in the plant. The data you collect can include times, temperatures, belt speeds, etc. You likely will also want to collect some microbial data on in-process and end product. Remember, you'll want a minimum of 3 replications of the study in your plant. Once you've gathered your data, you'll want to analyze it. You'll want to calculate means, as well as variance and standard deviation. Once you've done that analysis, you'll be able to answer the question if the critical limit was achieved. Documentation of the validation needs to be included in your food safety plan. This would include any data on safe harbors, scientific studies done by others, microbial challenge studies you've commissioned, and in-plant operational validation studies. Keep records of all this data to support the validation of your critical limit. The validation of a process preventive control needs to be reviewed by a preventive controls qualified individual or a process authority. Make sure you keep documentation of this review as well. Hopefully, this presentation has shed some light on the validation process. This final slide provides resources that can be helpful in learning more about validation.